Good morning, dear students. Today we can continue antibiotics and antimicrobial drugs. In last class we have dealt with penicillin and their derivatives and some other antibiotics which inhibit the cell wall synthesis in bacteria. Today we can study other antibiotics. The first among them is those which inhibit protein synthesis. Protein synthesis inhibitors, they act based on the difference between prokaryotic and eukaryotic ribosome structure. Protein synthesis inhibitors work at different stages of protein synthesis like initiation, elongation stage, which include either binding the amino acyl tRNA, or proofreading stage, or peptidyl transfer stage, or ribosomal translocation or termination process. So there are some antibiotics which interfere with the protein synthesis. First one is tigecycline. It will block the A site on the ribosome, preventing the binding of amino acyl tRNA. Then second group is aminoglycosides. They interfere with the proofreading process, which result in the increase in error rate, and therefore premature termination of protein synthesis will occur. Chloramphenicol, it will block the peptidyl transfer step of the elongation of the 50S ribosomal subunit. Then streptogramin, it causes the premature release of peptide chain. So among these antibiotics, chloramphenicol is an important one. It will block the peptidyl transfer step of the elongation in protein synthesis that is taking place at the 50S ribosomal subunit. Next is tetracycline. Tetracycline also inhibit protein synthesis in bacteria. It is a broad spectrum antibiotic. It can be used against many bacterial species. It is used to treat bacterial infections such as urinary tract infection, intestinal tract infection, eye infections, sexually transmitted diseases, periodontitis, that is gum infection, and other bacterial infections. So tetracycline is one of the most important antibiotic which is having a broad spectrum activity, which can be active against a large number of bacteria, causing a variety of infections. And tetracycline act by inhibiting the protein synthesis in the target bacteria. Then mode of action of tetracycline. Tetracycline inhibit bacterial growth by stopping protein synthesis. It can cross the membrane of the bacteria and accumulate in high concentration in cytoplasm. Tetracycline then bind with the 30th subunit and prevent mRNA from binding to the ribosome. In human cells, however, tetracycline does not accumulate in sufficient concentration to stop protein synthesis. So in bacterial cell, tetracycline, as they are able to cross the plasma membrane of the bacteria, they accumulate in sufficient concentration in the bacterial cell so as to bind with the 30th subunit of the ribosome, preventing mRNA from binding with the ribosome. But in human beings, tetracycline doesn't have any side effect because it is incapable of accumulating in larger concentration in human cells to prevent protein synthesis. Next antibiotic is erythromycin. It is produced by Streptomyces erythraeus. It inhibits microorganisms resistant to streptomycin and penicillin. So it is a broad spectrum antibiotic, and it is effective against those microorganisms which are resistant to streptomycin antibiotic as well as penicillin antibiotic. Erythromycin, it is effective against both gram-positive and gram-negative bacteria, and it inhibits protein synthesis by binding 50S ribosomal subunit. So erythromycin produced by Streptomyces, it inhibits both gram-positive and gram-negative bacteria by binding with the 50S ribosomal subunit. Next is those antibiotics which are capable of inhibiting nucleic acid synthesis in bacteria. The antibacterial actions of these drugs are a result of difference in the prokaryotic and eukaryotic enzymes involved in the nucleic acid synthesis. These antibiotics specifically target the enzyme RNA polymerase and DNA gyrase in bacteria. DNA gyrase enzyme, it is responsible for relaxing the tightly wound chromosomal DNA, thus allowing the DNA replication to proceed smoothly. But this antibiotic, this nucleic acid synthesize inhibiting antibiotics, it does not affect the DNA gyrase of humans. Antimicrobial rifampin, it binds with the DNA dependent RNA polymerase, thereby inhibiting the initiation of RNA transcription. So, these antibiotics inhibit nucleic acid synthesis, that is both the DNA synthesis and RNA synthesis of bacteria, but they never interfere with these RNA as well as DNA synthesis in human beings. So, they mainly inhibit two enzymes. First enzyme is DNA gyrase, which is responsible for unwinding the DNA during replication, and second enzyme is RNA polymerase, which is responsible for the production of mRNA from DNA. So, this nucleic acid synthesis inhibiting antibiotic affect these two enzymes in bacteria, thereby preventing their nucleic acid synthesis. An example is rifampin. It binds with the RNA polymerase enzyme in bacteria, inhibiting the synthesis of RNA. Second antibiotic is streptomycin. It is produced by Streptomyces griseus and it inhibits microorganisms resistant to sulfonamides and penicillin. It is effective against gram-negative bacteria and it is an aminoglycosidic antibiotic. So, it is an example of aminoglycosidic antibiotic. It is effective against gram-negative bacteria and it is effective against those bacteria which are resistant to sulfonamide as well as penicillin antibiotic, and it is produced by Streptomyces griseus. So, streptomycin, it inhibits nucleic acid synthesis in bacteria. Next is quinolones. Fluoroquinolones are synthetic antibacterial drugs with a broad spectrum of activity. They are used to treat urinary tract infections, nosocomial pneumonia, anthrax and plague, and they inhibit DNA synthesis by interfering with the coiling of DNA strands. So, they also, these quinolones are antibiotic which are capable of inhibiting nucleic acid synthesis in bacteria by binding with the or by interfering with the coiling of DNA strands. So, inhibiting nucleic acid synthesis, we have gone through 3 amino acid, rifampin, streptomycin and quinolones. Next is disruption of the plasma membrane. So, several types of antimicrobial drugs function by disrupting the plasma membrane of the bacteria. Plasma membrane has a variety of functions such as cell adhesion, ion conductivity, cell signaling, provide surface for attachment of various extracellular structures, and when this plasma membrane structure get disrupted, all these activities or all these structures get disrupted and it will result in rapid depolarization resulting in loss of membrane potential, leading to the inhibition of protein, DNA and RNA synthesis, and ultimately it will leads to the death of bacterial cell. So, the 3 main antibiotics which are responsible for disrupting the plasma membrane of bacteria include polymyxin, gramicidins and tyrosidins. So, polymyxins, they are effective against gram-negative bacteria. Gramicidins and tyrosidins, they are effective against gram-positive bacteria. So, polymyxins, gramicidins and tyrosidins, Tyrosidins are examples of antibiotics which will interfere with the disruption of plasma membrane in they will cause not the, they will result in the disruption of plasma membrane of bacteria, and by disrupting the plasma membrane, bacteria could not able to perform most of its metabolic activities, including synthesis of protein, RNA and DNA, as well as cell signaling, cell adhesion, etc. So ultimately it will lead to the death of bacteria. And examples of such antibiotics are polymyxins, gramicillin and tyrosidin. Polymyxins, it is effective against gram-negative bacteria. Gramicillins and tyrosidins are effective against gram-positive bacteria. Next is inhibiting essential metabolite synthesis. So there are antimetabolites. They are substances that compete with the with or inhibit the normal metabolic process, often by acting as an analog of essential metabolites. So antimetabolites are substances which mimic the essential metabolite, or they act as an analog for the essential metabolite, and antimetabolite can be used as an antibiotic. So three main type of antimetabolite antibiotics are there: antifolite, pyrimidine analogues and purine analogues. So pyrimidine analogues, they mimic the structure of pyrimidines and pyrimidine derivatives, and they disrupt the formation of pyrimidine, and consequently they disrupt DNA and RNA synthesis. So pyrimidine analogues are those antimetabolites which mimic pyrimidine like structure. So they will disrupt the formation of pyrimidines, and when the pyrimidine shortage will be there, then RNA and DNA synthesis will be affected. Next is purine analog. They mimic the structure of purines, and purines analog disrupt nucleic acid production. An example is azathioprine. Azathioprine, it is a main immunosuppressive substance that is widely used in transplants to control rejection reaction by inhibiting DNA synthesis in lymphocyte. So azathioprine, it is an immunosuppressive drug that is used to inhibit DNA synthesis in lymphocyte in those patients which is having transplants, to prevent the rejection reactions. So purine analog bind with the enzyme that is responsible for the production of purines, thereby inhibiting the synthesis of purines. Next is antifungal antibiotics. So antibiotics that are inhibiting bacteria is over. Then antifungal antibiotics. Antifungal antibiotics means those antibiotics which are effective against fungal infections. So example are nystatin and griseofulvin. Nystatin, it is used in the treatment of non-systemic fungal infections, and it is effective against fungi like yeast, candida and penicillium, aspergillus, etc. So antifungal antibiotic, first one is nystatin. It is effective against yeast, candida, penicillium, aspergillus, etc. and it is used in the therapy of non-systemic fungal infection. Second one is griseofulvin. Griseofulvin, it is obtained from Penicillium griseofulvin. So griseofulvin is an antifungal antibiotic which is originally produced by another fungus called Penicillium griseofulvin. It is used in the therapy of skin infections and systemic fungal infections. So systemic means deep-rooted fungal infections are there. So griseofulvin, it is used in the treatment of skin infections and systemic fungal infections. Next is antiviral chemotherapeutic agent. Example, interferons. Interferons are natural substances which are produced inside our body against viruses. And these interferons, they are very short lived and they are produced in very small amount. And interferons can be commercially produced by recombinant DNA technology, and they inhibit protein synthesis in these viruses. And an example is acycloguanosine, which is effective against herpes virus. Acycloguanosine is an antiviral chemotherapeutic agent which is effective against herpes virus. Amantadine, it is effective against influenza virus. So, acycloguanosine and amantadine, they are antiviral chemotherapeutic agents which are effective against herpes virus and influenza virus respectively. So, naturally occurring antiviral therapeutic agent is interferon. Next is anti-tumor antibiotics. Anthromycin group are anti-tumor antibiotic, and that group include several antibiotics like anthromycin, neothromycin, tomamycin. They are all potent anti-tumor antibiotics. So, they are effective against this uncontrolled growth of these tumor cells. So, they will prevent or they will cause the destruction of these tumor cells. Next is synthetic chemotherapeutic agents. Synthetic chemotherapeutic agents are those chemotherapeutic agents which do not occur naturally. Example is nitrofuran. It is effective against gram-positive, gram-negative bacteria, fungi and several protozoan infections. And second one is nalidixic acid. It is effective against gram-negative bacteria that is causing urinary tract infections. So, the synthetic chemotherapeutic agents are those agents which are artificially prepared. Example include nitrofuran and nalidixic acid. Nitrofuran is effective against a broad spectrum of microorganisms including both gram-positive and gram-negative bacteria, fungi and protozoa. And nalidixic acid, it is effective against gram-negative bacteria which are causing urinary tract infections. So, today we have dealt with different kinds of antibiotics. First is antibiotics which inhibit the protein synthesis. Second one is antibiotics which are capable of inhibiting this nucleic acid synthesis in bacteria. And the third is antibiotics which are disrupting the plasma membrane in bacteria. And finally, those inhibiting the essential metabolite synthesis in bacteria, especially purine and pyrimidine synthesis in bacteria. And finally, we have dealt with so many other antimicrobial drugs like antifungal antibiotics, antiviral chemotherapeutic agents, then anti-tumor antibiotics and synthetic chemotherapeutic agents. So, these are the references that I have made. First one is microbiology. It is a textbook written by Michael J. Pelzar, Chana and Noel Kirk. And these are the sources that I have searched from the net. So, this is all for today's class. Thank you.